Hi, this is Professor Tiffany Martin with Medical Microbiology, and we are starting Chapter 1. Chapter 1 is a lot of history, which is kind of fun and also kind of defines microbiology. So let's point out some important things on the PowerPoints with Chapter 1. Okay, so before we start talking about the history, we do need to define what is microbiology. And biology. Biology is the study of life, and micro is the study of small life. So these are going to include things, these are too small to be seen by the naked eye. It's going to include bacteria, viruses, protozoans, fungi, algae, and helminths. And helminths, if you don't know, are worms. Now, we've seen those monsters inside me videos. We know worms can get very large and we can see them with the naked eye, but some of their parts of their life cycle we cannot see, such as their eggs or larval stages. So that's why they are included in this category. Now, if they are too small to be seen by the naked eye, how do we know they were there? And the answer to that is for a long time, we did not know they were there. It wasn't until microscopes came along and people started looking at things did we discover these microorganisms. Now, microscopes came into were invented before the 1600s. They used glass lenses to pass light through that, and then they were able to magnify things, and they were able to see things they couldn't see before. So this is a picture of a simple microscope that would have been in use around the 1600s. It had a glass lens, and it actually had a place where you would mount your specimen. And a lot of people would make these and kind of keep them kind of just to display, and they'd come back and look at these specimens over and over again. Now, there was a gentleman living in this time period called Antony van Leeuwenhoek. He was Dutch. He was not a scientist, but he was actually a merchant. His job was to buy and sell fabrics. And he would use hand lenses or simple microscopes as part of his daily activities. He would look at the fibers of the cloth, and by magnifying them, he could really see the quality so that he wasn't paying more or less for quality merchandise, so he could get a good price for quality. So he would look at the fibers with these hand lenses. So that was his job, and he, again, wasn't a scientist, but he was a very curious person. And he started to look, since he liked looking at the hand lenses with fabric, it became kind of a hobby. He started looking at other things through these simple microscopes. Things he would look at would include insects, a lot of descriptions about the wing of a bee or an ant, and he would mount those up there. And so lots of different types of things. He was the first person to look at blood under a microscope. He was the first person to look at semen under a microscope. He was just very curious, and this was a big hobby for him. So one day he decided he wanted to look at pond water under a microscope. So scooped up some water, mounted it like he had blood and other things, and was expecting to see water larger. What he saw was all of these animals that he had never seen before. And there were things moving across and creeping across, very different. Now, Leeuwenhoek was a very detailed-oriented person, and he kept a lot of notes. We can actually look at his notes and figure out what he was seeing based on those notes. So he kept a lot of notes about what he was seeing, and we can see that he saw protozoans. Those were the ones that were moving, a lot of other different things under his microscopes. And because of his notes, because he saw something new, he was able to name it, and he called these things that he was seeing animalcules. He also used the term wee beasties to describe them, which is kind of fun. Now, we don't use these words anymore. We are now calling them microorganisms because they're too small to be seen, but he was the first person to see them. Now, he was a very private person, and so it took a while for the scientific community to kind of see this. Again, he wasn't part of the science, and he kept his objects kind of hidden from the public. He didn't want people to mess it up, and so he didn't share them very often. So it took a while, but based on the notes that he left behind, he is credited with being the first person to see these organisms under the microscope. Now, again, he did not invent microscopes. He just used them. Another thing I put on this PowerPoint, and we'll come back, is that his work inadvertently supported spontaneous generation. Another word for that is abiogenesis. So we're going to talk about that in a little bit, but I did want to put this in this PowerPoint so that as you're studying, you can come back to that. right, so what did he see? I'm going to let you review the next few slides and kind of familiarize yourself or refamiliarize yourself with some of these organisms. So take a few moments to do that, and then we'll come back and point out some highlights. Okay, so bacteria, he definitely saw some bacteria under this. Bacteria is a prokaryotic cell. Archaea is also a prokaryotic cell. We used to kind of lump these together in the same category, all as prokaryotes, but due to DNA technology and looking at their molecular structure of their DNA, we've actually found that these two organisms are extremely different, and so different that they've become its own category. So bacteria and archaea are separate. You've got bacteria, you have archaea, and then you have eukaryote, which is everything else, all of the eukaryotes for that. Prokaryotic cells, bacteria, we have a lot of diseases that are bacterial diseases. So we're going to talk a lot about that in chapter 3. Bacterial diseases include strep throat, meningitis, cholera, tetanus, gonorrhea. A lot of food poisoning is bacterial infections, staph infections, you've heard of that, staphylococcal infections. So there's lots of diseases caused by that. Archaea, we have not found any diseases that cause that, so we're not going to talk about that much in the course. Okay, and we will have chapter 3, which will talk about bacteria and their structures in detail. A fungus are now eukaryotic cells, so they have a nucleus versus prokaryotes without a nucleus. They do get food from other organisms, so they are not plants because they get food from other organisms. These include our molds and our yeasts. From that, lots of fungal diseases, yeast infections, different types of yeast infections. There's fungal pneumonias, nail infections caused by a fungus, very difficult. Lots of different respiratory diseases can be caused by fungus. And ringworm is actually a fungal infection. Okay, what else? Protozoans. These are probably Leeuwenhoek's wee beasties because they are capable of locomotion. Pseudopodia, cilia, or maybe flagellum. So definitely types of locomotion in this category. They are eukaryotic cells. Diseases caused by protozoans. Malaria is one. Brain-eating amoebas from that. So there are several different types of diseases caused by protozoans. So review those structures. Algae, no diseases caused by algae. Now there are some cases where an algae bloom in a lake or in the ocean can get the toxins produced by the algae can build up in fish or in shellfish. And by consuming those animals, the fish and the shellfish, we can take in a large dose of these toxins and can have some negative side effects from that. But there are no diseases from algae that directly affect us. They are photosynthetic. They are not plants. They are too simple for that. Lots of different types of those in this category. These are some single-celled algae called diatoms. And they have this crystallized kind of cell wall structure. We use diatoms in things that need friction, such as car polishes will contain that. We use other algae in our agar plates that we'll talk about in lab. So lots of things we can do with algae. Helmets and worms. Leeuwenhoek definitely saw these in his microscopes. These are all multicellular. Again, it's the smallest parts of their life cycle that make these microorganisms. Diseases, tapeworms, right? Pinworms, hookworms, lots of different diseases caused by worms. Viruses, Leeuwenhoek did not see. Why? Because they're too small. We were not able to see viruses until an electron microscope was invented. So instead of passing light through lenses, they were passing electrons, and that enabled us to increase the magnification. That didn't happen until the 1930s. So Leeuwenhoek didn't see them. However, we do know that they were there around the 1900s based on their behavior. So we'll have a whole chapter on viruses. Viral diseases, lots of them. COVID-19 is caused by a virus. Measles, mumps, think of your vaccinations. Chickenpox, herpes is a virus. HIV, lots of different diseases caused by viruses. Okay, so this was a brief introduction of microbiology. Again, review those PowerPoints so that you can know some facts about each one to get some idea. They will come back up during different types of case studies and other things that we'll do this semester. Okay, I'll see you in the next video. Thanks.